If you've been following the creation-evolution debate over the last year, and especially the human-chimp similarity issue, then you have surely not missed a series of articles by Casey Luskin at the Discovery Institute, with an initial article titled "Bombshell: New Research Overturns Claim that Humans and Chimps Differ by Only 1% of DNA." And as you can see in the title of this republication of the same article, this seems to have been actually their main story during the whole of 2025. So, as you understand, this article and this series of articles are challenging the commonly heard claim that we are 98 to 99% genetically similar to chimpanzees. And this is, as I said, a series of articles. So you can find, I think, most of the other articles in the series if you go on 1% Myth series on that tag. If you click that one, you get to this series of articles. Anyhow, if you've been following this issue, you have surely also not missed a response video by YouTuber Erika of Gutsick Gibbon with the title "Every Creationist Got This Wrong Because of Casey Luskin." So she made a really long video, 3 hours and 37 minutes, where she is providing a response and going into depth of this series of articles. And so my video is going to be a response to Erika's response video to Casey's initial series of articles. So the background to why this is coming out now, because there has actually been challenges to this number before, including based on an earlier paper in 2018 and calculations by Christian evolutionary biologist Richard Buggs, based on data from that paper, for example, and also earlier estimates. But why this is coming out now is because of an article in Nature earlier the same year, in April 2025, titled "Complete Sequencing of Ape Genomes." So what happened in this study is that for the first time we have complete sequences of some ape genomes, including the chimpanzee genome. And with complete I mean from the far end of each chromosome to the other far end in the regions called telomeres. So from the telomere region on one end of each chromosome to the other telomeric region. So that is also why these sequences are often called telomere to telomere, or sometimes abbreviated T2T. So this means that for the first time, since we have also the human telomere to telomere sequence since a couple of years ago, so now for the first time we can do a really good and comprehensive comparison between the human and chimpanzee genomes, or at least between one individual of humans and one individual of chimpanzees, because of course if we want to do a really comprehensive comparison, we need to do it based on a population of humans and chimpanzees to kind of get some kind of average similarity. But now we at least can do a very comprehensive similarity measure between two individuals. So the reason why this is so important is that the previous estimate of 98 to 99% was actually based on a part of our genome, the protein coding part, that is actually less than 3% of our genome. So naturally that doesn't give a good overall estimate of the similarity between humans and chimps. And the reason that we've been able to use only such a small part of the DNA to do the similarity has a couple of reasons. And one of them is that the rest of the genome has been really hard to assemble into long contiguous sequences, so it has been hard to compare. And the gene coding regions are less repetitive and they are easier to assemble and compare. But the other really big reason for that is that in the past we have actually viewed the other parts of the genome as mostly junk. And that has been put forth by famous atheists like Richard Dawkins, for example. And that has completely dominated the view in the field and made it so that we have not thought that we needed to even really look at those sequences. But that has completely changed over the last couple of decades, especially through big scientific projects like the ENCODE project that has gone through multiple phases, and they came out with the last results just in the last couple of days, where they show that actually most of this so-called junk DNA is actually active, and it's definitely not junk. So the view on these sequences has completely shifted as well, and that is some of the things that are going on in the background here as well, that also warrants that we actually need to look at all the sequences to get a proper view on the overall similarity here. So before I go into some more critical points regarding this video, I want to commend Erika for putting an enormous effort into investigating this issue, and also like reading through all of the material verbatim, not trying to put other words into Casey's mouth, etc., and also educating and explaining a lot of the details and the technical intricacy in this kind of comparisons. So let's get into why I'm doing this response myself. So Erika is pointing out some valid points, such as the omission of an earlier study in 2018 that was actually the first time we had chimpanzee genomes assembled without the human genome as a template, and also estimates of the similarity between humans and chimps based on data from that study by Richard Buggs. So she's claiming that Casey is making this new study appear as more new than it actually is. And there might be some merit to that claim. And she's also pointing out a case where Casey has been cutting out a part of a figure, probably to make the figure clearer to his point. And I think that is the main issues she's pointing out. But then she's also building a huge case around some rather technical aspects of how these similarities are done. She's pointing out that the similarity measure that we used before, the 98 to 99%, is one type of similarity, and the new similarity that we get based on the complete sequence of humans and chimp genomes, that these are actually two different ways of measuring. And that is completely true. And I don't think Casey has missed that point either, but he's not perhaps making it as clear as it could have been made, and that would have been helpful in his articles. But at the same time, I don't think that takes away any of the main argument that Casey is putting forth. And I think Erika is building a way too big case around that. And she's also portraying the whole situation in a very belittling and derogatory tone that I don't think is warranted. And also there are a number of points that she's making that is simply not accurate that I will also point out. So my hope with this whole video is simply to try to portray a more truthful picture of the whole situation. As a bystander, I will ultimately not try to defend the actions and choices that Casey Luskin has done. Those he have to, of course, answer to himself, and he's the only one that can do that. And I'm quite sure that he might do that at some point. I'm more as a bystander and third party, just want to point out some things that to me seem to be obvious mistakes and misrepresentations from Erika's side to give a more truthful picture. And also, before diving into this whole issue of human-chimp similarity, I think it's important to make it clear what relevance these actually have, because from a creationary viewpoint, the exact similarity measures actually don't mean that much, because we don't actually know what similarity measure we should expect if a creator created chimpanzees and humans, because we are actually very similar biologically. I mean, we eat bananas. Chimpanzees don't walk upright, but at least they can kind of move in a similar way as humans. We have a similar body plan, etc., etc. And chimpanzees also have a lot of intellectual capabilities that don't match the human ones. Far from that, but at least they get towards some part towards the human capabilities. So naturally we have a lot of biological similarities, and we don't know how much by necessity based on the structure and the architecture of the genome that we should expect to be similar. And that is because we don't still know completely how the genome works. We know a lot about how the genome works. Science has progressed enormously during the last couple of decades. But there's also a lot of things that we are only now starting to learn. And those are especially related to regulation of the genome. And the fact that we have only now in the last couple of years starting to learn certain things, that is exactly because of the same reason that we only now start to have complete genomes of humans and chimpanzees, that now we can span those very repetitive regions. Because it turns out that in those repetitive regions are a lot of sequences often called transposable elements, sometimes also called jumping genes, or a subclass of them are called endogenous retroviruses, and these are sequences that can jump around in the genome, although they might not do that all the time, but they can do occasionally. And we have started to see more and more in the last couple of years that these are actually involved a lot in the architecture of the genome, but also in the regulation of the genome. And only in the last year there was an article that highlighted some of the new, some new science regarding how the brain is developing based on stem cells between chimpanzees and humans. And this is so new so that we only have a preprint of it. So let's check this article out, or this preprint out. It's called 3D Epigenome Evolution Underlies Divergent Gene Regulatory Programs in Primate Neural Development. And we can look at just a quote here from the abstract. So this is a preprint, so it means it has not gone through peer review yet, but it is probably going through peer review right now, and we might see this ending up in some probably high-profile scientific journal later. And so what these authors say is, we identified human-specific epigenetic signatures, including cis regulatory regions and enhancer promoter interactions, and linked them to gene regulatory dynamics. Deep learning models reveal that complex regulatory grammar at cis regulatory regions, including transcription factor binding sites, local context, and higher order chromatin organization, underlie species and cell type specific differences. So they're talking here about species-specific regulation, specific for humans and chimpanzees, for example, that we're only now starting to see. And in the paper, they go into how a lot of this is driven by transposable elements specifically. So the point here is that we're only now starting to learn some of the details of how the genome is translated into the body. And this means that only when we start to have a really full picture of how that works, we can have a completely accurate understanding of how similar should chimpanzees and humans have to be based on the constraints from the architecture of the genome. And that means that we cannot really know what the creationary view would require in terms of similarity measures. But on the other hand, actually a too low similarity will actually cause a problem for evolutionists, since that will make it hard to explain how all of these differences have been able to arise in the available timeline. And that is perhaps one reason why evolutionists are so eager to portray chimpanzees and humans as very similar. But again, as I said, for a creationary worldview, it doesn't really matter that much. So the point of this video is not to debate trying to get this similarity as low as possible just to kind of strengthen the creationary worldview, because there's not really any point in that. The point is challenging this false notion that we are extremely similar to chimpanzees, between chimpanzees and humans, that evolutionists have been using. Because regardless if it doesn't matter, evolutionists are still using these numbers, since it has a psychological effect when they tell people that we are 98 to 99% similar. I mean, most people don't even understand what that means. And especially they don't understand how this number was obtained. Like, how did we calculate this number? For most people, it will just be a psychological effect of hearing a very high number, and people will just be intimidated by that if they are creationists perhaps, and they will just think it sounds scary or something like that. And because of that, it's very important that people understand the full picture. And just the fact that these 98 to 99% similarity is based just on less than 3% of our DNA means that it's not an accurate overall estimate to be communicated to the public, because people in the public wouldn't have guessed that that is the way this similarity measure have been made. So if you would use this number, you should actually provide it with a qualifier, like explain what type of similarity this is. Anyhow, Erika makes a huge point around this, and I think this is missing the point very much of the issue at hand. Anyhow, I will go through Erika's main claims one by one and answer them a little bit in more detail. And since Erika's video is very long, more than 3 hours and 37 minutes, I will not go through the whole video. Instead, I will go through her summary in the end. But I also will say that after reading this initial series of articles by Casey Luskin, I highly recommend that you watch Erika's video. It's worth watching, and it will also help you to follow the discussion here, and it will help you follow my response much better as well, because the summary is quite abbreviated, it's quite summarized, and doesn't go into a lot of depth here. So I recommend watching Erika's video and then coming back to watch my response. So let's get started. Alright, so let's go through the first section here.
Part 6. Summary of the stuff Casey got wrong and lied about. So let's go over the things that Casey got wrong in a nice, neat, little clippable section here at the end that you can kind of refer back to and quote if you feel like it. Number 1, he lies about the novel nature of this paper with relation to genome assembly and genome completion. This is specifically with relation to humans and chimpanzees, which is what his series of articles are about, but we were using non-humanized chimpanzee genomes and also pretty nearly complete chimpanzee genomes prior to this most recent study. So to imply that this is brand new and that's why the numbers are so groundbreaking is a lie.
Well, let's stop here. So if we go down and check what he's actually saying here regarding for the first time. So he's actually focusing on, at least in the latest edition of this article, he's focusing on that for the first time we now have the complete sequences of ape genomes. And as a result, for the first time, we can attempt a much more accurate assessment of the true degree of differences between the human and chimp genomes. There might have been other versions of this article before, but from what I could see in Erika's video, she actually had the same version of this paragraph as far as I could see. But another thing, it is true that Casey is not citing the Cronenberg paper, which was actually the first time they assembled the chimpanzee genome without using the human reference as a template when doing the assembly. And that in itself is a huge topic, maybe for another time. But actually in the Cronenberg paper, they also don't actually provide another metric than the protein coding gene similarity. Although this is the time when we should be really interested in having a kind of more overall similarity measure based on, for example, how much of the genome is possible to align towards each other between humans and chimps. So what had to happen instead is that Christian evolutionary biologist Richard Buggs had to go in and dig into the data and do some calculations, which he published in this blog post on his website, richardbuggs.com, how similar are human and chimpanzee genomes. And he has came to some estimates further down in his article. And you can check out the blog post if you want. But another thing, as I said, it's true that Casey did not cite the 2018 Cronenberg paper, and he didn't cite Richard Buggs' calculations based on that either. But you know what? Let's take a look at the Nature paper itself. If we go down here and look where they cite previous work, they actually also don't cite the Cronenberg paper. And I happen to know that because they actually cite the paper, but in another context, and it's number 59, we can search for it. And we see that it's citation 59. So if we search for 59 and try to find that publication, we find it further down in the article related to a very peripheral discussion, a rather technical one regarding inversions, etc., as part of a very, very long list of other citations. So how can it be that this huge article missed such a profound previous work? How can it be that this very, very long list of authors completely missed it, and also the peer reviewers missed it? At least we can say that Casey is not the only one who missed to properly cite that previous work. Okay, let's continue with number 2.
2, Casey tells us that this new percent similarity replaces the old percent similarity. We have understood since the beginning of comparative genomics that alignment numbers, percent similarity in protein coding regions, percent similarity across the whole genome just looking at single nucleotide polymorphisms, and percent similarity looking at the whole genome with those S and Ps plus indels included are 4 separate methods of comparing genomes, and they are not even the only 4 methods of comparing genomes. Likewise, we have had estimates since 2005, but really good estimates since 2013 for the alignment percent similarity between humans and chimpanzees, in addition to the protein coding, SNPs only across the whole genome, and SNPs plus indels across the entire genome. So, to imply that a new alignment number, which matches quite nicely with older estimates, is replacing an old sequence identity number, which the new estimates also map very closely with, is a lie.
You know what? You're completely wrong here, and your argument here is completely irrelevant. It is true that there are separate ways of calculating the similarity, and that in the new paper they have multiple actually ways of calculating the similarity, and the protein similarity is quite similar to as before. But if you look at what Casey is discussing, he's not actually discussing this topic in terms of either of these specific ways of measuring. He's discussing this in terms of an overall estimate of the similarity. And you can see that very clearly if we go down into the citations of how this similarity has been used. Because it has not been touted as a protein sequence similarity only. Rather, we have seen citations like, we share more than 98% of our DNA and almost all of our genes with our closest living relative, the chimpanzee. And that is from a Nature, paper or a news article or something like that. Again, in National Review, we have about 90% of our DNA is identical to that of chimpanzees. We have here, most studies indicate when genomic regions are compared between chimpanzees and humans, they share about 98.5% sequence identity. And perhaps this is the only one where they are clear about that it's about the genomic regions, which I understand that they mean the protein coding gene regions. But the other ones as well. Humans and chimps share a surprising 98.8% of their DNA. American Museum of Natural History. Humans share about 99% of our DNA with chimpanzees, making them our closest living relatives in science. Chimps, humans, 96%. So you see here how the numbers here are not used qualified with any of these different ways of measuring the similarity. Rather, they are touted as an overall estimate, like regardless of measurement technique. So going into technical details about which metric is replacing which one or not replacing totally misses the point here and is completely irrelevant to the discussion here. So what I mean is that Casey is not discussing his argument in that level of detail. He's discussing this overall measurement that has been put forward. And as these citations show, this is also how it has been used by others before. So that is the topic of Casey's articles, obviously, at least from what I can see. And of course, it's interesting and it can be helpful to look into those more detailed various ways of measuring the similarity. And it could be helpful to discuss that. And of course, Casey's articles could be probably improved by providing more of that detail and go into depth of that discussion. But he's not doing that here, and it's outside of the argument that he's making. And another thing that shows that actually others also tend to think that we have a new situation now when we have the telomere-to-telomere genome and can actually look into, have a more proper picture of the similarities here, is a quote that Casey found in an older version of the preprint of this article that says, the oft-quoted statistic of 99% sequence identity between chimpanzee and humans holds for most of the genome when considering single nucleotide variants. However, comparisons of T to T genomes, that is the complete ones from telomere to telomere, the far ends of each chromosome end, suggest a much more nuanced estimate. And we can go in and check the preprint here ourselves. We have here the citation here, quote. So the point here is that they also see that this 99% sequence identity is not a good overall estimate, and they don't have a problem contrasting that towards the new, much more nuanced picture. So this also shows that this is a new situation we have now that we have the full telomere-to-telomere genome. So this actually speaks both to Erika's claim 1 and 2. Okay, so let's continue with number 3.
That leads me to number 3, which is really a combination of 1 and 2. Casey obfuscates that we've had estimates for all 4 methods of comparison in question for many years. It's true that Richard Buggs was one of the first ones to propose the alignment numbers in 2008, but since 2013, as I've showed you, we've had estimates for all of those methods of comparison, and that's an important point since Casey goes on to imply that something nefarious is at play and is responsible for why the alignment numbers aren't front and center as this big brand new difference between humans and chimpanzees. I know that Casey is actually aware of the difference between those 4 methods of comparison, and he's aware that there is a history behind those 4 methods of comparison, which means the fact that he hasn't edited for clarity those initial articles means he's lying again.
Yeah, so Richard Buggs was the one proposing the alignment number, and he was also the one that had to go dig out those numbers in the 2018 paper. And that in itself raises some questions. So, as I said in the previous section, Casey's argument is taking issue with using the number as an overall estimate without qualifications regarding measurement technique used. So these metrics and ways of comparison are really not very relevant here. Of course, it would be helpful to discuss in terms of them to give kind of more understanding of the intricate details of how we actually are comparing these sequences. But as you have seen, it has been Christian scientists who have been proposing and digging up those alignment numbers from the start. The secularists have been dragging their feet behind them when it comes to providing those numbers. And I'm happy that they are providing them now. But apparently it took Christian authors to get them to do that. And it's true that we have had those numbers for some time. And it would have been good if Casey had cited that and made that clearer. But those details, as I have said also, are not really relevant to his claim in this matter. And what's more, it is actually the first time that we have complete chimpanzee genomes and can do a complete comprehensive comparison. So even though we have had estimates earlier, this is the first time that we can do it properly without using assumptions or need to just estimate the number. We can go in and measure to the best of our techniques. And that also, like as the previous quote showed in that reprint, the quote that they removed, that there are others that also would think that the 99% similarity is not really showing the whole picture here because of what we now have in this complete telomere-to-telomere sequence. Okay, so let's do the rest of claim 3.
Since this is the Casey obfuscates portion or number of the summary at the end, I'm going to add that he also edited a figure to get rid of inconvenient information for an argument he was trying to make and ignored data immediately on top of a data point that he was reporting that he found was beneficial, but then he obviously didn't report the data point that he found to be inconvenient. There seems to be a trend here.
Right. So this I don't have to answer myself since Casey already answered it shortly, I think, after Erika's video came out. So if you go in on the fact check article, the second article in Casey's series, and scroll down a bit here, you can see that he has updated and added an author's note, July 8, 2025. I post here the full chart from figure 3.12 of the supplemental data from you et al. 2025 because some people are claiming that I originally omitted data from the chart because it somehow refuted my argument. No, that's false. The only reason I posted only the human-ape genome comparison, the top 8 curves, was for simplification purposes because that was the only portion of the chart that was relevant to my argument. The intraspecific genetic differences, the bottom 5 curves, are irrelevant. My argument is not that human-ape genetic differences necessarily refute evolution. It's that evolutionists who for decades have claimed humans are only 1% genetically different from apes are wrong. The top 8 curves demonstrate my point very clearly. There was and is no data in the chart that negatively affected my argument, and I always linked to the supplemental data where people could see the complete data set if they wish. There's nothing to hide here. In fact, I already extensively addressed objections regarding intraspecific genetic differences here before recent critics started commenting. So I will leave this at that. So let's also go through claim number 5, which is closely related to the previous one though.
If you want humans and chimps to go from 98.8 to 95, you're also going to have to drop humans from 99.9 to anywhere from 87 to 94%. The differences between humans and chimpanzees are by and large analogous, owing themselves to large-scale duplications, large-scale deletions, large-scale insertions, and large-scale inversions. These types of mutations, when they occur in the non-functional portions of the DNA, can occur and accumulate very rapidly, which is why the differences exist within species and also between closely related species like chimps and bonobos or Sumatran and Bornean orangutans. It's also why we shouldn't use it as our method for determining percent similarity between organisms. Well, that and the fact that if you do choose to use it, racists will be all over it, erroneously so. Number 6.
Well, I think it's very silly, the whole thing, like how she's purporting this as some kind of argument against what Casey is trying to say. He is claiming that it's erroneous to propose 98 to 99% as an overall estimate of the human-chimp similarity. And if changing it changes the similarity between humans and humans and other animals, so be it. As I said in the beginning, we actually don't know yet completely what those changes mean because we don't still have a complete picture of how the genome works. So the exact numbers don't actually matter that much, or at least we don't know how much they matter. But it's wrong to go around and tell that we are 98 to 99% similar in our DNA without qualification, without telling how this similarity was done. And as you could see in the citations in the beginning, that is what has been done. It has been put forth as an overall estimation that people who are not into science would always assume that it's, of course, a measure of the whole DNA, because that's what they are saying. Our DNA is 98 to 99% similar, is what some citations say. And that is wrong. And this whole discussion of other ways of measuring and the consequences of that, that's details. And it's irrelevant to the argument, because nobody has been trying to say that these similarity measures are what determines who we are more closely related to than compared to others. I mean, of course, we expect those numbers to say something, but we don't know. We don't know what those changes mean. Maybe there are only a certain type of differences that are the very important ones that determine the differences between main types of animals. We don't know because we don't have a full picture of how the genome works, as I've been showing some examples of earlier in this video. So I think it's just a very silly argument. It's irrelevant to the argument.
6, Casey's articles are completely bereft of any additional comparisons of organisms that he accepts are related. Yeah, this is the same problem that Tompkins had. It's funny, there sure is a through line through all of these creationists in their inability to actually do good science when it comes to humans being apes, closely related to other apes. I wonder why that is. Casey includes 0 comparisons of anything other than a human to one of the apes, and that...
Yeah, maybe because that was not part of his argument, as he has been trying to clarify a number of times.
That's really important because those other comparisons ground and contextualize the percent similarity between humans and chimps.
Yeah, if we think the similarities actually mean something, if we have a very fixed understanding of what those similarities mean. And I think the evolutionists have that because they're measuring the distance in those similarities. But from a creationary view, it's not a given what those differences actually mean. So nobody with a creationary worldview are actually trying to argue that those differences are what matters, because we don't know that at this point. So Erika is arguing out of her evolutionary worldview, and because of that, she's upset that Casey is coming with a different worldview, as far as I understand. And yeah, what a mess.
Regardless of which method we're using. I already included the differences between some of the other ape pairs, chimps and bonobos, and Sumatran and Bornean orangutans, but it's worth mentioning that tigers and domestic house cats are like 95% similar looking across their genome on the alignable regions, but that drops to like 84% using Casey's methodology. Likewise, rats and mice are like 80 to 85% similar when looking at the alignable regions, but that drops to like 70% or lower if you want to use Casey's methodology. I don't doubt that Casey would think that cats and tigers or rats and mice are actually related.
We'll go more into the rats and mice a little bit later as part of when Erika is looking into the comparative genome viewer.
The reason he doesn't include them is because that makes it so abundantly clear that the differences between humans and chimps, no matter how you categorize them, aren't very big.
Well, and that was not the argument either. The argument was that it's wrong to say that we're 98 to 99% similar to chimpanzees without qualification.
So I want to take one more detour and do a cool little visual representation for folks out there who may be visual learners, as I am. NIH and NCBI has this really cool genome viewer, comparative genome viewer, where you can take a species and an assembly and compare it to another species and assembly and see how they actually map to one another. This is a super cool little tool. As you can see, we're looking at two humans here. We've got, I believe these are our two haplotypes. We've got HD002 on top and the telomere to telomere primary, I think, on the bottom. And they're not identical, right? You can see there are mismatches all over the place, things ending up where they ostensibly shouldn't be. And if you want, you can show only forward alignments or only reverse alignments here on the bottom. Our reverse alignments are ones where we've seen an inversion, I believe, so...
Yeah, this is actually a kind of interesting point, because earlier in the video, Erika was claiming that inversions were probably not included in the similarity measure, and because of that it would be kind of affected. But clearly inversions are included here. So the fact that she has not gone back and edited her earlier parts of the video when she was realizing it here raises perhaps some questions if we should use the same standards.
Gone on between two humans, and all of this adds up to that percent difference when we're looking at an alignment as a whole. Here's our human and chimpanzee primary. I believe these both also come from the Grade A Genomes paper, and you can see that we've got a lot of stuff ending up where it ostensibly shouldn't be, but more than that, we have a whole lot of reverse alignments, so things that have sort of inverted in on themselves in addition. This is obviously way less similar than what we saw with a human to a human. Chimp versus bonobo, or rather bonobo versus chimp, falls somewhere in between the two, right? Like it's less similar than what we saw with human and human, but it's more similar than what we saw with human and chimp. That maps with our understanding of how phylogeny works. Now, my understanding is that we're looking at the portions of the genome that can be aligned here. We're not looking at, like, alignment numbers themselves, because where would the lines go? I want to show you a comparison. Here's our human versus chimpanzee again, or rather a chimp on the top versus human on the bottom. I want to draw your attention to one other thing, which is how human chromosome 2 is a fusion of chimp chromosomes 12 and 13. That's kind of weird, isn't it? The synteny, how this synteny looks. Why is chimpanzee chromosome 12 and 13 like aligning onto human chromosome 2 and all of 12 is in reverse so that they can fuse telomere to telomere? That's not what I want to show you. What I want you to do is soak in the differences that you can see between the human and the chimp. Again, this is the stuff that, to my understanding, is already aligned. This is excluding non-alignable regions, I think, because I don't know where the lines would go.
I want to show another thing here as well, actually. If we go in here on the human to chimpanzee comparison that Erika is showing just now, she was showing some of these things. Actually, there are more things here as well. You can include the non-best placed alignments, and that also shows the problems and challenges about trying to align things. You actually have multiple options on how to align things here. Another thing is that you can also show like the minimum alignment size that should be included in the diagram here. So that can be also kind of interesting, and that means that we understand that there are some assumptions going into this alignment. Another thing is that you can actually zoom in by clicking on these chromosomes, and that can be quite fun actually. Let's back out here. Let's continue.
And then I want you to compare that to this. This is the rat versus the mouse, right? We're looking at a rat here on the top, the Norwegian rat, and a house mouse here on the bottom.
Yep.
And I hope you can see visually now just how much more different rats are to mice as compared to humans and chimpanzees. Not only do they have a lot of sequence identity differences, right, like a greater amount than humans do to chimpanzees, but their genome is like completely scrambled compared to one another. Everything is seemingly in the wrong place. There are all of these massive inversions. And like most creationists would tell you that these two are related by genetics and by morphology, but that these two are not, and obviously so. I hope you can see that that's stupid and dumb. But
Erika, I think you know, it's interesting that you bring this example up particularly, because I think you know something about animals. I think you know that mice and rats, that they have a different generation time than humans and chimpanzees, right? Humans have a generation time of something just roughly around 25 years. But is that the case for mice and rats? One can do a little bit of Googling or asking some chat GPT or whatever, and you will see that the generation time for especially mice and also rats is much shorter. So they're producing offspring like instead of years, they are counting it in months, in actually much fewer months than humans are counting the years as well. So it's actually sometimes up to over 100 times shorter generation time. And you probably also know that it's during each generation that you often have these crossing over events and chromosomal restructuring events. That means that this is exactly what we would expect, or this is nothing extraordinary or surprising in any way, because they will have just a lot more generations in the same time frame, and that will mean a lot more chromosomal restructuring, at least some 100 times more or something like that. And it's actually known also that the rat and mice genomes are very prone to restructuring because of very active transposable elements and things like that. And I don't think this is unknown to you, Erika, so I'm a little bit surprised that you bring up this example actually.
All of this, I feel the need just to point out one more time for everybody in the back, with relation to humans and chimpanzees, and this is something that I checked with the authors, our understanding of their relationship hasn't changed. Humans and chimps are still more closely related than any is to a gorilla, they're more closely related than rats are to mice, and that's problematic for somebody like Casey Luskin, because that relationship holds no matter which method you use. We get the same phylogeny every single time. This is why what's going to end up happening is what we got a little taste of in this very last article. They're going to...
Yeah, well, if you count relatedness only using phylogeny, and if that is your assumption that everything is related, and then you force everything into a phylogeny, then of course you will get that. And I'm not surprised. Why would that change? That is not surprising to anybody here. So I don't see your point here.
Resort to the types of differences. And that's fine, but that has no bearing on common ancestry. So when you utilize the usual 99% number or the 96% number, and some creationist comes at you and says, didn't you see the new study? Humans and chimps are actually only 85% similar. You're going to say this in response. Actually, the information in that study with relation to humans and chimpanzees isn't new. That study actually supports the notion that humans and chimpanzees are more closely related to one another than either is to a gorilla, and that 85% number is actually a different method of comparison as
Yes, but it's also a much better estimate of an overall similarity of the DNA. If you're going to say, give an overall number without qualification. The best is, of course, to never give an overall number without qualification and telling like how you did the estimation, because as you say, it's a very complicated topic. But why have a lot of the popular science and scientific outlets everywhere been putting forth this number without qualification as an overall estimate of the DNA similarity. And that's the point, and that's the thing Casey's series of articles was taking issue with, as said.
Compared to the 99% number, both of which are methods we've known for a long time. That second method is actually pretty flawed. Did you know that if you use that with two humans, they could end up being only 87% similar?
And so what?
7% for two humans versus 85% for humans and chimps sure doesn't seem like that big of a difference, does it? Cut.
And so what does this matter? It's only if we assume that this number actually means something, if this is kind of the ultimate source of information about how similar we are. And we don't know that ultimately, because we don't have a full picture of how the genome works, and we don't have a full picture of exactly which parts are more important than others. Of course we know a lot, but we don't have a full picture of that. And then it's really hard to determine what we should expect in terms of similarity here. So, as said, again, as far as I can see, and as Casey is actually clarifying himself now seemingly multiple times, the issue he was, the thing he was taking issue with was the false notion that 98 to 99% is a good overall estimate, unqualified, for the human-chimp similarity.
Print wrap. Now, after looking into this pretty extensively, I feel pretty confident in just using the roughly 99% number for the percent similarity between humans and chimps. I think we should always be clear with our words and what we're actually comparing there. And
yeah, and most news and science outlets haven't been that clear with that, right? So I'm happy if you are doing that, but others haven't been that.
If someone pushes back and asks for the entire genome, nucleotide by nucleotide, that raw alignment, including S and Ps, we can talk about that too. We just have to remember where that lands other pairs of organisms. That has to be a part of the conversation.
Yeah, and that is especially important in an evolutionary worldview and paradigm where these differences are much more important or have to be important, which they don't necessarily need to be in a creationary worldview. And that is, I think, the source, again, for a lot of the confusion here.
Especially with relation to evolution and creationism. Good thing I've been doing that.
Yeah, so not at all very much related to evolution and creationism, because evolution and creationism have very different expectations on what similarities we should expect, as I explained. The creationary worldview doesn't actually set a specific number or specify a certain number that or a significance to that number, at least until we completely understand the genome and understand which parts of the genome are influencing the differences more than others, etc. And I think we're only just starting to learn some of the things there.
This entire time, and essentially everybody else I know who's a science communicator here on YouTube has as well. Casey has not slain an icon of evolution here because it hasn't been an icon of evolution for a very long time. The myth...
Well, I don't think Casey has slain an icon of evolution. I think the icon of evolution has been slain in multiple parts. As you say, it started actually already in the Cronenberg paper, but then those authors were, as said, not publishing the alignment numbers themselves. Those had to be dug out by a Christian scientist, and now in this article we get a much more complete picture. And now I'm happy that they publish the alignment numbers, maybe because of efforts by scientists such as Richard Buggs. So the science is starting to show up here, but of course it's different ways of comparing. But as I said, if we're talking about an overall estimate, unqualified estimate of the similarity, then I think it's quite obvious that the alignment number is a much more accurate estimate. But as you say as well, I mean, if you want to use other metrics and tell and explain how you obtained those metrics, I'm fine with that as well.
Myth of 1% has been busted by the conventional scientific community for a really long time. There's actually articles with that title. It's a perfectly fine number to use as long as you're clear on what you're talking about. The truth is, you can't
I think that's right.
home in on a single universally applicable perfect number that everyone agrees upon for the
Exactly, and that is why we should not use the 98 to 99% number as an unqualified overall number, as a lot of scientific journals and news outlets have been doing all over the place.
for the percent similarity between any two organisms, because there's so many different ways to compare them.
Very good point. There's so many different ways of comparing them, and because of that we should not go around with this single universal number 98 to 99%, because it's very misleading, especially for the public who don't have the slightest on how this number was obtained.
For my money, I'm just going to stick with the methodology that the entirety of the genetics and genomics community uses, and that's 96 to 99% for the similarity between humans and chimpanzees. It's also 99.9% and up for humans and humans. And it's 95% for tigers and house cats, and about 85% for rats and mice, and so on and so forth.
Yeah, different animals have different generation times and different characteristics when it comes to chromosomal stability, etc. Nothing surprising there.
It may be that in 50 years, the entire genetics community is just using the alignment number, but they're not using it now, and I trust them way more than I trust any of these creationist goobers on the subject. If they change to alignment number, I'm happy with doing that, because remember, everything's going to change proportionally. The phylogenies will still exist and be just as problematic as they always have been for the creationists, and they're going to create some really interesting problems for themselves.
Yeah, again, not really accurate there. These similarity measures are not really that relevant to a creationary worldview. So, yeah.
Given that chimps and bonobos only hash out as 1 to 2% more different under this new alignment method than humans and chimps, how are you going to explain that one under a design model, guys? Or in 6,000 years?
Yeah, so what's the problem here, actually? The similarities here are important for evolutionists, because that's the only way. You have to assume that all the changes have arisen through evolutionary processes at some point. Creationists don't need to assume that. So what is the problem really here?
My prediction, though, is that they're just going to keep presenting all the numbers. They might put them all up front and center in the introduction. That would probably make the creationists more happy, at least. But I don't think that we're going to get the alignment number only, because remember, it's not really telling us anything, or certainly not the whole story. And when that happens, the entire conventional scientific community will just be like, cool, business as usual, and the creationists will rage and seethe and maul over the...
Yeah, because they can just continue to drum out their evolutionary worldview. So for them, they're not threatened in that sense, because they have decided and they have the support of the whole secular scientific community, whatever the results will show. I mean, and that tells something about how the evolutionary theory is not a falsifiable model, because it's an assumption and it's a worldview, and you will always interpret everything in that worldview, regardless of what the results say anyway.
The fact that we're still seeing the 99% number, because it's still really relevant for protein coding regions and whole genome with S&Ps. But humans and chimpanzees being 96 to 99% similar to one another is something that just doesn't sit well with creationists in general. It's why it consumed Jeffrey Tompkins.
I don't think you really understand the creationary view on this properly.
And why it's consuming Casey Luskin. They have to get that number down because they can't stand it. They don't want to be that...
Well, we could say that we don't stand lies or misleading information. that is giving people a false sense of something that is not actually true, regardless of what that means.
That's similar to a chimpanzee, but my dude, even if you are 99% chimp, you're 100% ape, because no matter how you hash out these percent similarities, humans
in your worldview,
nest firmly within hominidae, and you can't get around that. One thing that I try to emphasize on this channel is that, again, that doesn't make humans any less cool. It doesn't make our story any less epic, and it definitely doesn't preclude your ability to be religious generally or to be a Christian. To quote one of my favorite creationists, the data are the data. So that's it. That's our deep dive into Casey Luskin's lying about this subject and his bungling of some aspects of it. I don't like Casey Luskin very much.
Well, as I think I've shown, I think calling this lying, I think it's a far overreach. There are some things that could have been made clearer. There are some papers that could have been cited that a very major Nature publication did also not cite. Yeah, and then there was a figure that Casey had edited, which he has now since fixed and added a note about as we went into. So I don't see that there's a lot of substance here. Yeah, and of course, one of the bigger issues that Erika is also bringing up is the proportion of like the other similarities between other pairs of humans and other pairs of animals. And as I said, that totally misses the point, because that doesn't matter in a creationary worldview, so it's quite irrelevant to the issue at hand, especially for us creationists. It's a bigger issue perhaps for evolutionists.
I didn't like him before, I really don't like him now. Here's what I'd like to see from him, and here's what I need to see to stop picking on him. I need to see not a retraction necessarily, but at least an edit on some of those past articles to clarify the things that he got wrong and the half-truths that he told, because a half-truth is a half-lie. Moving forward, I want to see Casey put his money where his mouth is and provide the context for the percent similarity looking at those alignments plus S and Ps, since he wants everybody else to do the same when we're talking about the percent similarity for protein coding regions or whole genome plus S and Ps or whatever. That also means, for my role in all of this, he needs to acknowledge that humans and chimps are more closely related than either is to a gorilla, and that the phylogeny has maintained through all of these years, as well as these numbers not being new. I need to see a lot of revising. He doesn't...
Well, Erika, I think it would then also correspondingly be nice if you would provide with some edits here regarding your misrepresentation of what Casey is trying to say, and trying to put it as though he was arguing for the fact that these similarity measures actually matter to the creation evolutionary debate, because clearly, just as my view, it's quite obvious that he also views that as irrelevant to the issue, or to the creation evolution issue, and that what he was taking issue with was the falsehood of providing a single unqualified overall number for the human-chimp similarity, which is based only on less than 3% of our DNA. So I want to cover another topic as well that Erika is mentioning earlier in the video, and that is regarding the functionality of these parts of the DNA that have been traditionally been regarded as junk, which is especially those very repetitive sequences, and which has true projects like the ENCODE project in multiple phases have started to be regarded to have functionality. The ENCODE project has reported a number of like 80%, at least 80% is functional of these sequences, and the leader of the ENCODE project thinks this is only to go up. Anyways, Erika is taking issue with this, and I want to comment a little bit on that.
So let's talk about the functionality of the human genome really quick, and how we know that no genome is 100% functional. I think this is pretty important. This is the one.
Yeah, so I don't know that creationists in general are claiming that 100% of the genome is functional. The argument is that it's not largely junk as it has been claimed in the past by people like Richard Dawkins.
1-2 knockout punch that I use, although if you want this more in depth, you can go to friend of the channel Creation Myths, Dan's channel, where he actually uses the literature to break down how we know what portions of the human genome are functional and how we know what the upper limit of functionality will be. I think he does a really good job at it. I'm not going to do that here. I might throw the numbers up here though, just so you can see. I'm going to focus on what I think is the simplest explanation and the easiest way to explain to creationists that we know genomes aren't 100% functional. And that's this paper all the way from 2004, right? So like there's no excuse for not knowing about this thing. It's called Megabase Deletions of Gene Deserts Result in Viable Mice. That says it all, but let me break it down. Megabase deletions, this means millions of base pairs are being deleted from gene deserts or regions of the genome not thought to be functional, result in viable mice, result in mice that can live and reproduce, and their reproduced offspring are themselves viable. This means that the whole genome isn't functional, because a lot of time what creationists will do is they'll appeal to regions of the genome perhaps not being functional in the life of an adult, but then they argue maybe it's important for development or for development specifically in utero. This blows that to smithereens. We'll read the abstract real quick. Right at the beginning, the functional importance of the roughly 98% of mammalian genomes not corresponding to protein coding sequences remains largely undetermined. Here we show that large-scale deletions of the non-coding DNA, referred to as gene deserts, can be well tolerated by an organism. We deleted 2 large non-coding intervals, 1,511 kilobases and 845 kilobases in length from the mouse genome. Viable mice, homozygous for the deletions, were generated and were indistinguishable from wild-type littermates with regard to morphology, reproductive fitness, growth, longevity, and a variety of parameters assaying general homeostasis. Further detailed analysis of the expression of multiple genes bracketing the deletions revealed only minor expression differences in homozygous deletion and in wild-type mice. Together, the 2 deleted segments harbor 1,243 non-coding sequences conserved between humans and rodents, so like this would be the same in humans, more than 100 base pairs, 70% identity. Some of the deleted sequences might encode for functions unidentified in our screens. Nonetheless, these studies further support the existence of potentially disposable DNA in the genomes of mammals.
Well, you have it right there, right? You had it right there in the abstract. Some of the deleted sequences might encode for functions unidentified in our screen, because that's exactly what I was going to ask you. Because do you really think that the criterion of viable and producing viable offspring is a good screen for functionality overall? I would think that is a very low bar, because there might be a lot of functionality that doesn't directly affect viability and the ability to produce offspring. So I think this doesn't say very much at all, actually, to be honest. As said, I don't know of creationists that argue that 100% of the human DNA is functional. The argument is that it's not largely junk at all, so we can't just dismiss it out of the comparison. And we actually don't have a full picture of the functionality of these sequences yet. We can't rule out that there's functionality there that we don't know. To the contrary, we've started to see and find more and more indications of functionality, actually especially in different cell types. And it's only now that we start to have single cell techniques, and we can do actually also sequencing and a very comprehensive mapping of sequencing and gene expression in all of the different cell types. So I think the jury is definitely still out on how much of the human DNA is actually functional, but it's clear that we cannot just dismiss large part of it as junk. And again, still, I would not be surprised if there's some amount of it being junk. but probably not 98 or like 96% or something like that. So to summarize, I think I've already summarized the main problems I see here, but all in all, I don't see that Erika's claims are coming with a lot of substance. There are some things that could have been made clearer. There are some articles that could have been cited, and it's good that Casey now has clarified about that picture that was edited. But overall, I think the main confusion is coming from the fact that Erika has misinterpreted what Casey is actually arguing for, that he would somehow argue or assume that these similarities actually are highly relevant to figuring out if we are closely related or not, which is quite clearly not what he's trying to argue for, but rather he was arguing for the fact that 98 to 99% is it's very wrong as an overall estimate of DNA similarity without any qualification, and that is exactly how it has been used in a lot of occasions. And so I think the very belittling and derogatory tone that Erika has towards Casey is very unnecessary. And I also think it's a shame because apart from those parts, there's a lot of merit to the video. It's helpful and pedagogic in many ways, but it's unfortunate since Erika is missing the point of what Casey is trying to say, and then she's framing him as a deliberate liar, which is quite sad. So that's it. I think that will be more than enough of my comments to this whole issue. As I've been saying, I'm a third party mostly about this, so Casey is the one that ultimately can answer to what his intentions, etc., with his articles. But I hope that my comments have been able to clear out some things that might not have been obvious to everybody. And as you see, this is the first article on this channel. I don't really know what the future holds. I have some ideas about doing deep dives into the origins issue. So if you're interested in this topic, feel free to subscribe and share this video, especially to people you think might be helped by it. Then we'll see what happens with this channel in the future. So in the meantime, have a good time and see you next time.